Welcome back, AP Stat students. I hope you had a wonderful winter break. In semester 1, we talked about descriptive statistics, and in semester 2, we will talk about inferential statistics. So the whole purpose of second semester is, what can a sample tell us about our population? So we will start chapter 7. Chapter 7 is called sampling distributions. In statistics, we gather data about a population. If possible, we will gather data about every person in the population. This is known as a census. If we cannot gather data from the entire population, we gather data from a sample of the population. We then use that to help us understand what is happening with the population itself. So if we look at the figure to the right, we collect data from a representative sample, and then our whole goal is to make an inference about the population. So the question that students ask me all the time is that sample representative of the population? So we learned in chapter 4 about the various different types of samples, like a simple random sample, the SRS, or a stratified random sample, cluster sample. So that was really important to pick the best possible sample so that we minimize the bias, because we can't eliminate bias, but we want to minimize it so that we can make an inference about the population. When we talk about sampling distributions, we have a couple of vocabulary words. The first one is called a parameter. A parameter is a number that describes some characteristic of the population. In statistical practice, the value of a parameter is usually unknown because we cannot examine the entire population. A statistic is a number that describes some characteristic of a sample, specifically a random sample. From the population. The value of a statistic can be computed directly from the sample data. We often use the statistic to estimate an unknown parameter. So just remember this: statistics come from samples, whereas parameters come from populations. And now we're going to distinguish the differences in notation. Just a heads up, if on the AP exam you freeze and you just forget the notation, it's always better to write things out. So whenever you're in doubt, write it out. If you don't remember the symbol, say the word. So we are going to use the Greek letter, well, it's the Greek letter mu, so it's the U, with the longer tail in front. So we're going to use that Greek letter mu for the mean of the population. We are going to use sigma for the standard deviation of the population. This is stuff that we learned first semester. So that's basically like a little O with a kind of like a hat, and that represents the standard deviation of the population. So this is for the population, and this is for quantitative or numerical data. We are going to use for the samples X bar to represent the mean. So hopefully you remember this, of the sample of the population, and we're going to use S sub X to represent the standard deviation of the sample, and that's for quantitative variables. When we're interested in our means and standard deviation. When we have categorical variables, we are talking about our proportions or our percentages. And so for our population, we are going to use the letter P to represent a proportion of the population. So this is for the population, and this is for the sample. And we're going to use p hat, similar to the y hat we used when we were talking about predicted values. So p hat is used to estimate the unknown parameter p. So the parameter p is the population proportion, p hat is the sample proportion. And over here I put a little note. We also use percentiles, quartiles, medians, but the ones listed above are the most commonly used. So you can use median and IQR, but that's used when data is skewed. The ones that I have in our table are the ones that we are going to use. They're commonly used. Let's look at an example. Identify each bold underlined number as a parameter or a statistic. Use the appropriate notation to identify each. Determine if the variable is categorical or quantitative. Number 1. On Tuesday, the bottles of Arizona iced tea filled in the plant were supposed to contain, on average, 20 ounces of iced tea. Quality control inspectors sampled 50 bottles at random from the day's production. These bottles contained an average of 19.6 ounces of iced tea. So the type of variable here that we are talking about will be quantitative because it's numerical. So it's quantitative. And the units measured are ounces of tea. Our parameter, so we're talking about our population, would be mu, and it would be 20 ounces, because if you look, the bottles filled in the plant were supposed to contain, so our mean, mu, is 20 ounces, and therefore the sample was 50 bottles chosen at random, and the average was 19.6. So that average, X bar, is 19.6 ounces. Okay. Number 2, on a New York to Denver flight, 8% of the 125 passengers were selected for random security screening before boarding. According to the Transportation Security Association, 10% of the passengers at this airport are chosen for random screening. So here, the type of variable is categorical. And the categories are either you're screened before boarding, or not screened before boarding. Our parameter is 10% of the passengers at this airport. So that's our population. So our P, So our P is equal to, and when we're dealing with percentages and stats, we always want to change those to decimals so we can do calculations. So P is equal to 0.10, and our statistic would be P hat, and that's 8% of the 125 passengers chosen at random. So make sure you put the proper notation. Here we go. Great. How can a statistic be an accurate estimate of a parameter? After all, different random samples would produce different values of the statistic. So we analyze what we call sampling variability. The value of a statistic varies in repeated random sampling. To make sense of sampling variability, we ask what would happen if we took many samples. So in this diagram, you have the population, and then you have all these samples. So what if we took all these samples, we got these statistics? Look at all the values from the sample, and then what would that tell us? So basically the question mark just says, hey, we're looking at all the values. What is it going to tell us about the sample? There are actually 3 distinct distributions involved when we sample repeatedly and measure a variable of interest. So here, this is the big idea of 7.1a. We have On the left, we have a population distribution. So that basically talks about everything. The population distribution gives the values of the variable for all the individuals in the population. So in this particular example, it's talking about chips. So the population is there's 200 chips, and they're either blue or red. So that's the color. So that's, and our variable is color. That's our population distribution. Okay, so from this population distribution, we are going to take many samples. If you look over here, this is a simple random sample of size 20. And then you have another sample, and yet another sample, and so on, all of size 20. So what we do is we graph that information. So when we took a simple random sample of size 20, we had 12 that were blue and 8 that were red. And we're only looking at the red in the middle column. So the first one, these are all your statistics right here from the p hat. So the p hat is 8 out of 20 were red for that top one. The second one, 11 out of 20 were red. But all of those are your statistics. So each of these is a statistic because we gathered that from a sample. Then on the very far right is what is known as a sampling distribution. So what it does is it takes all those sample data that we have in the center or in the middle of our diagram, and we plot that. So for instance, point 40 would be right here, point 55 would be right here, point 65 would be right here, and this is just a few, this is just 3 of them. We're looking at, this actually is 500 simple random sample. And here is the approximate sampling distribution. And if you look at it, it's pretty symmetrical. So some of the things that we can talk about is that it's symmetric. It's unimodal because there's one peak. And the peak is like at 0.50. It's somewhat bell-shaped. The center is at 0.5. And the spread could be between, if you think that 0.15 is an outlier, the spread could be 0.25 to 0.80, and potential outlier at 0.15. So that's what we can do with your sample data, the distribution of the sample data. You can make a sampling distribution, gather all that information. So basically, how do the samples and sampling distribution relate to the population distribution? That's our goal. That's what we're trying to answer. So let's look at here. The distribution of the sample, so the second column, shows the values of the variable for all the individuals in the sample. And the sampling distribution, the one on the far right, shows the statistic values from all the possible samples of the same size from the population. So those are all samples of size 20 from the population. And our parameter in this case is the proportion of red chips in the population. So our parameter is 0.50. And those are our notes for 7.1a.